So I'm Rainer Engelhardt. I'm acting as an introducing speaker and as well as sort of moderator, trying to keep everybody on time here. I'm the Assistant Deputy Minister for the Infectious Disease Prevention Control Branch with the Public Health Agency of Canada, if you didn't know that from before. However, you may not know that CHVI, the Canadian HIV Vaccine Initiative, is a brainchild, really, of the Public Health Agency. So that makes me particularly interested to be here and introducing you. So I want to welcome you to this event, which will highlight some of the achievements of the Canadian HIV Vaccine Initiative's research developments, along with a keynote address that we'll have this morning. The CHVI is certainly an innovative partnership, fits very well with the theme of the CAHR conference here. And it's a partnership. It's a partnership among several Government of Canada departments, and that includes the Public Health Agency, Health Canada, Canadian Institutes of Health Research, Canadian International Development Agency, and Industry Canada, and bringing them all together to the table in a joint focus and enterprise, I can tell you, was not necessarily easy years ago. It was like herding cats, but it's come together in a very effective interplay and partnership. The other element to that partnership is the Bill and Melinda Gates Foundation, global leader, as you know, in funding HIV research, and vaccine research in particular. And they are a strong player in that partnership, a strong and contributing player. I'd like to also maybe take the opportunity just right now to thank the CAHR conference organizers for ensuring a strong focus on HIV vaccines research as part of this conference, and simply for putting, again, a very promising conference together. Also want to acknowledge our Alliance Coordinating Office for the CHVI that are here, that have helped to put this session together and are helping with moving the agenda forward altogether on HIV research. And for those of you who don't know, the Alliance Coordinating Office, it's housed at one of the collaborating centers in Canada, in this case, the International Center for Infectious Diseases in Winnipeg. So this is not about me introducing here. We have a list of speakers. They're on the side, listed on the side. Our speakers are sitting at the right. They are going to be appearing in order. We've got 4 principal investigators and closing with a keynote speaker. Our principal investigators are going to give thumbnail highlight sketches of the work that they're doing under the CHVI funding, and then allowing a little bit more time for our keynote speaker at the end. So I'll introduce everybody individually as they come up. So our first principal investigator is Ken Rosenthal. He's the Division Director of Molecular Medicine at McMaster Immunology Research Center, McMaster University, and head of the Viral Vaccine Division. His project team has partners in South Africa, and its aims are to identify factors in breast milk that protect infants from HIV infection and other inflammatory responses of the infant gut, following different feeding practices that may increase susceptibility to HIV vaccine. So with that, HIV infection, excuse me, with that, Ken, if you would please.
Thanks, Ron.
It's a pleasure.
Appreciate it. Thank you. Good morning. Good morning, everyone. Okay, this is my conflict of interest disclosure slide, title slide. Our study, just to make this fairly quickly, what I'm going to try to do is very quickly give you a bit of an overview of what our study involves. We're calling our study the infant study for innate factors associated with nursing transmission. The study of HIV-exposed uninfected individuals has really contributed to our understanding of factors that might be involved in protection against HIV. And exposed uninfected infants, as a study group, offer some advantages over some of the other cohorts of exposed uninfected individuals. Notably, the mother and the infant are always together, and the mother, obviously, if transmission occurs, would be transmitting the virus to the infant. Our interest in looking at breast milk and HIV was sparked by studies that showed a very interesting paradox. The paradox is that HIV-infected mothers who exclusively nurse their babies protect the babies against infection relative to mothers who used mixed feeding, which is paradoxical because the infants are actually consuming more virus on a daily basis for months, if not years, compared to the mixed-fed infants. And this led us to come up with a hypothesis that perhaps short-lived innate factors in breast milk, which are major constituents of breast milk, are playing a key role in this protection. The hypotheses that underpin our study are listed here, essentially that innate immunomodulatory and antiviral factors in human milk protect infants against HIV infection. We have in fact identified there are a number of factors in milk that have anti-HIV activity, but we recently identified a novel factor, soluble toll-like receptor 2, which is in high level in breast milk, and it actually will inhibit HIV infection directly as well as inhibit immune activation by the virus. But other hypotheses are listed here that in the mixed-fed infants, perhaps exposure to contaminants are inducing inflammation in the infant's gut. Also that increased immune activation in the mixed breastfed infant will alter innate and adaptive immune responses to normal pediatric vaccines. And we have social scientists in our group who will look at breastfeeding behaviors as well as whether mothers would enroll their infants in vaccine trials. So in a sense, the study will prepare cohorts of infants in Africa for vaccine trials. The study design is listed here. It's essentially a prospective observational cohort study. We're going to be recruiting 500 positive HIV-infected mothers and their infants, and we'll have control groups of uninfected mothers as well as mothers who bottle feed or formula feed their infants. The populations that we're going to be studying are going to be cohorts in South Africa as well as in Nigeria at the hospitals listed here. We believe this is going to give us a unique opportunity to expand our understanding of the factors that would interfere with breastfeeding and willingness of mothers to enroll their infants in trials. So we're going to be looking at enablers and barriers to various breastfeeding behaviors. The labs involved in the study are located in Nigeria, at the University of Cape Town, as well as in Canada, both at McMaster and at Manitoba. We're going to be doing the proteome of human milk and the lipidome of human milk. We'll be training people through the CAPTAIN network, or the Canada-Africa Prevention Trials Network. And basically the proposal is a multidisciplinary approach to HIV vaccine research that exploits the human model of mother-to-child exposure to try to identify correlates of protection. And we're going to have behavioral studies there to look at breastfeeding behaviors and attitudes towards infant feeding. Thank you.
Thank you, Ken. That's fascinating and very promising. I think we'll all be looking forward to those results. So our next speaker is Dr. Jerry Zaharadas, who is assistant professor at McGill University and an infectious disease specialist, clinical scientist, also at the Jewish General Hospital in Montreal. So Jerry, please.
Morning. Thank you to the organizers and especially to Mark Weinberg for giving me the opportunity to speak for our group. These are my disclosures. Our team is called the Botswana Canada AIDS Vaccine Discovery Partnership. The team leaders are Mark Weinberg and Rosemary Musunda of Botswana. This is the makeup of our team. It's composed of virologists, immunologists, clinician scientists, and people really who are working at the interface of all those disciplines, and is comprised of members of McGill University affiliated institutions, Simon Fraser University, University of Amsterdam, and of course a significant portion of the team is composed of people from the Botswana-Harvard Partnership, which is an offshoot of work that was pioneered by Max Essex at the Harvard AIDS Institute. Really, the question we're looking at, the space we want to look at, is the space between the development of autologous neutralizing antibodies specific to HIV envelope and broadly neutralizing antibodies. And as many of you will know, there's been an incredible explosion of work on broadly neutralizing antibodies. But we thought one area, and this was the brainchild of Jamie Scott, she thought the one area that we should look at was cross-neutralizing antibodies, and that really hadn't been looked at in the kind of detail we're going to be talking about, and understanding how you go from autologous to heterologous or cross-neutralizing antibodies and eventually to broadly neutralizing antibodies would be a key question in vaccine design. So this is just a diagram of the panoply of monoclonals that have been described. Almost 100 now, when we started with 4 or 5. There's a number of them that have incredible potency and breadth, and that finally are starting to yield some information about what an immunogen should look like. And I'd refer you to work by Leo Stamatatos and Bill Schief and Bart Haynes, just published in the last couple of weeks, in terms of how to get around the problem of stimulating those unmutated germ cell BCRs and getting them to move towards producing broadly neutralizing antibodies. So we're taking a similar approach to what others have taken in terms of isolating broadly neutralizing monoclonals. But specifically, we're going to be looking at patients that are recently infected, trying to understand what their transmitted founder virus looks like. And we just don't understand what that bottleneck is. And then trying to understand, as immune responses come up later, how specific pathways are selected and why certain antibody responses are favored. We're going to be designing immunogens and B-cell selection reagents to try to fish out some of these B cells, and eventually, again, specifically targeting the cross-neutralizing antibodies, trying to figure out what an immunogen should look like specific to transmitted founder viruses. So these are the questions we'll be asking. Are there common features in transmitted founder viruses? What envelope epitopes do these antibodies target? And how exactly does virus and antibody response co-evolve? These are some of our objectives. So we want to look at transmitted founder virus envelope. We want to create a panel of Botswanan-specific reagents. Besides neutralizing antibodies, we also want to look at ADCC and non-neutralizing antibodies. And really, we want to define when cross-neutralizing antibodies come up, how they interact with viruses within a cluster and among clusters. And eventually, we'd like to understand how we can build an immunogen to try to stimulate some of these B-cell responses. This is the area where in Botswana where our partners have worked. So a rural village called Mochudi. They've done some beautiful work in terms of looking at viral linkages within the community and understanding transmission clusters. Taking a page from this work, we're going to be developing resources within the southern part. And we'll be looking at staging, trying to figure out where people are in their infection and trying to recruit people in these earlier stages before feedback stage, at or before feedback stage 5. This is what our algorithm will look like. Once we triage people to recent infection, we will try to recruit them to our study. And there's been some progress with regards to the clinical team on the ground, and we're now awaiting regulatory approval, which is, I think, forthcoming. We've made some steps forward in the lab in terms of building reagents that are going to be specific to Botswana or adapted to Botswana. And finally, this is what our timeline looks like, sort of perhaps a little bit forward-looking, but assuming regulatory approval comes forth, we're going to be starting to build a lot of these Botswanan-specific reagents. And I thank you for your attention, and once again, I thank our team members and the organizers.
Thank you very much, Jerry, for that brief, as it is, expose to some innovative immunogen design goals. We're going to move from that to social design innovation. Now, Mark Brockman, Dr. Mark Brockman, is an associate scientist at the BC Centre for Excellence in HIV/AIDS, associate professor at Simon Fraser University. He's examining barriers to engaging young people in HIV vaccine trials. His study team is collaborating with investigators in South Africa. The results are anticipated to inform social and biomedical aims of future HIV vaccine studies involving minors. Please.
Good morning. It's a pleasure to be here and be part of this symposium. So I'll briefly describe the project that we're doing as part of the vaccine initiative. I myself am a biomedical researcher, so I'm a little bit of a transition, I think, from between the last couple of speakers and Peter, who will be coming up next. So we're really taking a slightly different tact than the last couple of presentations, and we're focusing more on a population, and we're trying to develop a multidisciplinary approach to look at this population of adolescents in South Africa. So I think everyone in the room will understand that South Africa is really a key site for the HIV/AIDS epidemic internationally. It's estimated there's approximately 6 million infected individuals in South Africa. But I think more relevant to our project is that the prevalence rate among adolescents and young adults is incredibly high in this population. So our project is based in 2 sites in South Africa, Durban on the southeast coast, as well as Soweto, a suburb of Johannesburg. They're indicated on this map. And we're really focusing on adolescents and young adults, so individuals between the ages of 15 and 24 years old. Young South African women are particularly at high risk of HIV infection, and this is largely due to a number of different social factors. It's estimated about 30% of them have children before the age of 20. They're often in relationships with older male partners that don't have a known HIV status. And approximately 21% of females in South Africa between the ages of 20 and 24 are living with HIV currently. So this is clearly a high-risk population. And therefore, adolescents and young adults are a key target population for eventual HIV vaccines and clearly prevention efforts that are ongoing. So really the problem that we're trying to address is that the risk of HIV acquisition, clinical outcome following infection is influenced by, we think, an array of social, behavioral, as well as biological determinants. However, there's not a lot of high quality population-based linked data sets for these types of populations where you have very good social behavioral information, very good clinical information, as well as very good biological or biomedical data. And so we're really trying to fill this niche and having a multidisciplinary team approaching each of these aspects individually and linked in a linked form in order to fill this gap. So what we've anticipated as goals and objectives is really shown on this slide as fourfold. We'll develop additional infrastructure and site capacity to do multidisciplinary research at these 2 locations in South Africa, both of which are actually very good in one or several aspects of the project, but we're going to extend their capacity in various ways. We have a component of the project that's really ethics and led by Jeremy Snyder from Simon Fraser University, as well as people at HAVIG that you'll hear about, I think, from Peter next, the HIV/AIDS vaccine ethics group at the University of KwaZulu-Natal, engaging with the community and stakeholders and looking at the ethics of not only vaccine delivery in these populations, but informed consent, parental consent, et cetera. A group of social scientists that are looking at behavioral issues, as well as a group of biomedical scientists that are looking at the biology of HIV and other sexually transmitted infections in this population. So just briefly to kind of give you an outline of the team, again, there's the 4 main highlights, a group of clinicians essentially working in both sites building cohorts, a group based both in Canada as well as South Africa looking at the ethics, as well as an international group looking at the social and behavioral aspects of infection, as well as a group looking at the biomedical issues. And in general, the plan is shown here. We're in the process of receiving ethics to begin enrollment, which we hope will begin in the fall of 2013. So later in this year, we'll have about 300 HIV negative individuals, 100 HIV positive adolescents and young adults, and we'll be following them for about 2 to 3 years to finish out the project. We really hope that as a result of this project that will improve the stakeholder knowledge as well as community engagement and interactions at these sites, as well as to have a much better understanding of the epidemic from a various numbers of aspects. And in particular, from a linked standpoint, we hope to look at host genetics, sexually transmitted infections in these populations, and how behavioral and biological factors are contributing to risk. So for that, I'll thank you, and I'll just acknowledge again all of the number of co-investigators and PIs on this project. Thanks.
Thank you, Mark. Also fascinating from the population and social perspective. We're going to continue in that theme in general with Dr. Peter Newman, Professor, Faculty of Social Work, University of Toronto, Canada's Research Chair in Health and Social Justice. He's launching an expert interdisciplinary social sciences team in Canada, India, and South Africa. His CHVI team, this is a CHVI project, clearly. The research program addresses key challenges in ensuring fully informed consent among HIV vaccine trial participants and in engaging the preferences and concerns of vulnerable community end users in order to bridge the science to practice gap across HIV vaccine development. So, Peter, please.
Thank you very much, and thank you to the organizers, Canadian HIV Vaccine Initiative, and the Alliance Coordinating Office for having me here, and also engaging in this, I think, really great endeavor. I really do acknowledge my colleagues very strongly. I've written some somewhat caustic stuff out there in terms of the lack of engagement of social science and a lot of biomedical and clinical research around HIV. And these folks are doing really fabulous work in terms of really meaningfully engaging behavioral and social science. And I think if that's one outcome to come out of this project, I think it's great. Okay, so this is my team. This is our up-and-coming website. And a lot of people, but so at Graham Lindegar, the PI in South Africa, from HAVEG, as was mentioned, University of KwaZulu-Natal, and Dr. Chakrapani is the PI in India, with whom I've been working for about 10 years in terms of various kinds of HIV prevention there. So just basically, our main research objectives are twofold, and one is to enhance comprehension of key concepts for informed consent, and the second is to advance the implementation science of future HIV vaccines. In terms of the first, we're starting in a more qualitative, open-ended place to explore indigenous understandings and mental models, how people understand HIV vaccines, and also, very importantly, clinical trials. In India, we're having a very hard time with, this is with a low SES community of MSM, men who have sex with men in India, of even getting at the concept of placebo. And it's not that people are stupid, it's just that they don't get it and they don't really buy why. Why do we need to have placebo control? So it's very important to get people on board. Out of this basic work, we're going to, qualitative, we're going to develop an intervention that's an enhanced informed consent. And we're going to try to use that to optimize comprehension of 2 or 3 concepts in the existing form. And then we're going to compare that to the existing to see if we've gotten a more meaningful understanding of some of the very key concepts in HIV vaccine trials. And you know, we don't, but it looks like placebo and perhaps vaccine-induced seropositivity are a couple that are quite difficult nuts to crack. And the second aspect of the project is we're looking at also, this is even in preclinical research, the importance of looking at the preferences and needs and understandings of communities for whom we're developing these vaccines. Because if a vaccine does come along in another decade or 2 or whatever it is, and people are not on board with it, it's going to delay much further uptake and coverage in terms of the populations, and particularly in HIV, when we're talking about some of the most marginalized communities around the globe, it's really important to engage these communities in the research process, to feel part of the endeavor and to feel happy about things moving forward. And we're using fairly advanced techniques with a very multidisciplinary team in terms of social work, psychology, economics, and using a tablet-based survey using discrete choice modeling to look at preferences for future vaccines, and also what would be the trade-offs for people in terms of, obviously we can't engineer an ideal vaccine, so what might be the implications? How do we prevent, what do we need to help to prepare people for? If we understand that, you know, some concern about side effects is very powerful, then we need to start learning about how to do that. So just briefly, in India, where I had a head start because I had an earlier CAHR grant looking at participation in willingness to participate in HIV vaccine trials, we've gotten some work done which is paving the way towards the early work of the team grant, and so this is close one recently, and we've really advanced looking at willingness to participate from a much more social ecological perspective, that it's not just an individual's decision, that families and communities weigh very, very heavily into these decisions. And so it's really trying to turn the model upside down a little bit about how do we look at factors in willingness to participate. In South Africa, we've just completed subcontracts and we're in the ethics approval process with our various partners. And we held a really great cross-team meeting, which is part of the beauty of this grant, is really leveraging South-to-South collaboration of Indian, South African investigators, and also my Thai colleagues as well. So just to remember that each year of delay in testing and rollout of future vaccines and other new prevention technologies, rectal microbicides, PrEP, and the like, there are millions of new infections every year that are on the line. And so that the idea is to prepare using social research throughout the development process of new technologies, and that we have so much to gain. And I think it's really a 1 + 1 = 3 synergy when social and biological, behavioral sciences work together. Thank you very much.
So thank you, Peter, and thank you to all of our 4 PIs here who are giving you a flavor of really of what the whole CHVI research engagement is about. I mean, showing that the type of research that's carried out is a very broad spectrum, from biomedical, pharmaceutically related research to clinical trial studies to intervention research. So with that, I have my honor to introduce our keynote speaker, Dr. Larry Corey, who is president and director of the Fred Hutchinson Cancer Research Center and a professor of medicine, laboratory medicine at the University of Washington. He's also the principal investigator of the U.S. HIV Vaccine Trials Network. During his presentation, Dr. Corey will provide us with a global perspective on HIV vaccine trials. And we're pretty good for being on time, so there may be an opportunity at the end of Dr. Corey's presentation to have some questions of himself or our 4 PIs. So Dr. Corey.
Pleasure. My pleasure. So thank you. It's always wonderful to drive up to Vancouver and see, you know, how it puts Seattle to shame in a lot of ways. But anyway, it's a lovely city and I'm honored to be here. I guess there's my financial conflict of interest. I'm clean in HIV. All right, that goes on my position as head of the HVTN. So I'll start with sort of the traditional why you should be listening to us. The need for an HIV vaccine hasn't changed. In the US, we continue to have an epidemic even though we've had antiretrovirals for a long time. HIV is spreading in Asia, and despite the treatment as a means of prevention, and which is, you know, has some traction and is starting to make a dent. If we really talk about what the goal is, me as a grandfather, I'd love for my granddaughters to grow up in a world without AIDS, and like I grew up, and that means that our zero has to be not reduced from 50,000 cases in the United States to 25,000 cases, but, you know, really to 100 cases or less than 100 cases. So there's no precedent of a sexually transmitted infection, let alone an asymptomatically acquired infection that is transmitted asymptomatically that can be controlled without a vaccine. So we are all for the biomedical strategies, but, you know, in the end, if we are going to control this, we need to develop a vaccine. And I think we are on mark, and I think RV144 helps us to say that the vaccine really should do what other vaccines do, i.e. prevent infection. So I'm going to start with the Thai study, because this trial provided the test of concept that we actually could prevent acquisition of infection. The nihilism, I think, of many about HIV vaccines, I think, has been sort of taken away by data. There's a major shift from attempting to reduce post-acquisition viral load to reducing the frequency of acquisition. And essentially, the surprising finding, and I think one of the concepts that I'll tell you, is that we in science have lots of biases, and the biases are often wrong. And in HIV vaccines, they've been wrong for most of the time. And where this efficacy trial has led us, has been to an area of the genome that was essentially ignored. And even when there were a few articles, they were really not given much credibility as being important, and I think is leading us to an area of attack that we need to think about as a lead. Just to remind you that this was a study that used, if you go back to a lot of the early work of this vector with LVAC. In fact, for some of the very first studies in phase 1, I was the lead author on, and I was not ever that impressed with the immunogenicity that this vector had. And then it was given with recombinant GP120, which in and of itself had already been shown not to be effective in increasing acquisition among both MSM in the United States as well as IV drug users in Thailand. And hence, when there were many skeptics, and many of us were surprised when you put the two together, you actually saw this event, which was a very large study among heterosexuals with a very low infection rate, in which the overall vaccine efficacy on the modified intent to treat curve was right here at 31%. But the interesting thing about the curve is, number one, is it separates pretty early, even after the second dose of vaccine, even before the GP120. There is sort of a peak area of large separation, and then it continues over time. So most of us were surprised by the result. By modern assays, the vaccine was not very immunogenic, induced no CD8 responses, 5 or 10% of people. It did induce lymphoproliferation in a high frequency, and it induced CD4 T cell responses by ICS assays. Hadn't performed very well in non-human primate challenge studies, and of course one component of it was not effective at all. This is too much data, it just says what I just said. If it's real, and if so, how did it work? And I think the last year and a half has been really one of the neat things about science, and really one of the great, I think, accomplishments of the HIV field, in which a group of people got together internationally, formed a series of committees, went off and with a wide variety of really thought leaders and labs, put together a comprehensive package to sort of ask the question, can we come up with immune correlates of the response, and can we try and understand what we had here? And I think that out of the sort of remarkable collaboration of validating assays, finding assays in which the biostatisticians said, hey, they were good enough and representative enough and could discern between placebo and vaccine, such that we actually could take these precious sera of looking at a case control study of who got infected, who didn't get infected, who received vaccine and was protected versus who received vaccine and did not. We have really seen, I think, one of the great things in science, certainly in the HIV field, emerge in the last 3 or 4 years of all these labs getting together in a very rigorous way and starting to weave together a story that I'll go over, which gives us some, I think certainly to me, increases the veracity of the accuracy of the RV144 trial and gives us some leads as to where we should go, that we certainly have some novel, what we'll call correlates of risk or correlates of protection, that, whether they are true mechanistic correlates underlying the true pathogenesis of infection, I think, is still in doubt, and it's pretty naive to think that just one aspect of this virus is susceptible enough so that we're going to, just hone in on one region and that will give us the holy grail. But the data are certainly, I think, interesting and encouraging. So the first lead was, frankly, just a linear peptide analysis that the Army did very early to actually look at the immune response of linear peptides across envelope to ask, what was induced by the vaccine. And that was actually the first clue to look at the V2 region, because you can compare the immune responses between vaccine and placebo. And also, interestingly, not illustrated on this slide, was the immune responses between vaccinated and HIV infected. That there was a concentration of immune responses to the linear peptides, both in the antibody in serum as well as in PBMCs to the V2 region. And when you actually did overlapping peptides, it was into the sort of the constant region of the V2 region. And that actually was the sentinel thing in sort of saying, well, maybe we should actually look at antibodies and other things to V2 in the case control studies. So this team that was led by Bart Haynes and Peter Gilbert, who's the marvelous statistician at the HVTN, who is just really brilliant, and Julie McElrath, who has set up the HVTN lab, who is really a wonderful scientist also, as well as Susan Zollner-Pasner, who had written about V2. And, because it was sort of a variable region, people had sort of said, what emerged from that was, the ability to take a sort of what is a constant part of a variable region called the GP70 of the V1, V2 protein, and put that on a murine leukemia virus scaffold, and show that actually reacted to vaccine sera, had a very good, tight assay. And frankly, when you actually fractionated the sera and looked at people who had vaccines who had high levels, medium levels, and low levels of antibody to the V1, V2 loop, you see people who had high levels were markedly protected, low levels less well protected. Actually, the protection rate between these two groups is over 70%. The other interesting aspect of this was that there was actually negative protection in another assay. So when you took binding antibodies, just sticking GP120 on a plate, and then looked at serum IgA antibodies, again, that was looked at, mainly because A, you could do it, and B, that they were looking at binding antibodies to IgG and IgM, and they threw in IgA. And lo and behold, there was sort of an inverse correlation. Those who had very high levels of IgA antibodies actually had higher rates of acquisition. Whoops, if I can go back. Higher rates of acquisition. The good news was among placebo recipients or among vaccinated persons who didn't acquire HIV, there was no evidence that this was an enhancement. But that the people who had essentially low levels of IgA antibodies were protected. Those who had high didn't have increased amount responses, but had the same responses as placebo. And there was some really interesting data that there was an association between low levels of IgA and good ADCC. There was good association between low levels of IgA and reasonable levels of neutralizing antibodies and low levels of IgA and reasonable CD4 responses, suggesting that this might be a blocking effect. And we'll come back to that because actually there have been mechanistic studies to actually show that these IgA antibodies essentially block the ability of IgG antibodies to do ADCC, maybe even neutralization. So there's a blocking antibody effect here. The other interesting thing is when the time course of these antibodies were done, one found that after vaccination, one induced reasonably high levels of antibody, but there was a pretty rapid decay. And that has been the case with one of the problems in all of HIV vaccines with all recombinant proteins to date, has been the durability. A HIV envelope in and of itself is not that durable as an immunogen. You took Alvac and put in a rabies envelope protein, you get rabies antibodies and neutralizing antibodies for a long time. You put Alvac protein and put in HIV envelope, you get, it just lasts for a little while and goes away. HIV envelope is not a very immunogenic protein, and that's one of the issues that we are facing, and whether that's due to the germline or other factors or a combination of factors is probably both. So conceptually, we throw out the hypothesis that protection is mediated by some aspects of IgG-V2 antibodies. They appear not to be neutralizing, at least by the way we do neutralization, but they appear to be functionally active. And whether that functional active is in reducing the efficiency of HIV infection in some virion capture way, or it increases the efficient clearance via ADCC, or elimination of HIV-infected CD4 T cells by ADCC, or efficient clearance by Fc receptor binding. All of these are viable hypotheses about, quote, function of what we generally call non-neutralizing antibodies. How would we show increased risk of an infection mediated by high titers of IgA envelope binding antibodies? Is that these antibodies essentially cause an inefficient clearance of HIV clusters, or inhibition of ADC mediated by IgG-F2 antibody by blocking the Fc receptor. And actually, Bart Haynes and his group actually have done some of that data with Georgia Tamaros to actually show that it's occurring. Now, okay, that's reasonable data. But I think probably the data that is emerging of most interest that I think leads us continuing to work on the V2 loop antibody is what Peter Gilbert and company have done, is what's called the Sieve analysis. Now, the Sieve analysis is really one in which you actually use the genetics of the virus to actually compare and ask the question, is there sort of immune pressure such that the genetic sequences of the virus that are infected breakthroughs, i.e. people who got vaccinated who broke through, do they differ in their genotype versus placebos? And the good news about that is that it is, in a randomized trial, a true difference between the vaccine and the placebo group. All the stuff in the antibody is between those who got vaccinated and got infected versus not, and sort of in a statistical sense can have a selection bias about who genetically responds or not. This is essentially more pure with respect to a correlation. So they looked at the fact, is there a Sieve effect? If a Sieve effect is affected in regions of V2 to which the RV144 vaccine-directed antibodies, it may suggest that these antibodies really had a role in protection, and might be useful in predicting protection. And the long and the short of it is the answer to this is yes, and it looks at 2 immune responses to the V1, V2 region. And some work has gone on from that to actually show that monoclonal antibodies to these responses, to these regions, are actually functional, which markedly enhances sort of the mechanistic underpinnings of this protection. So this complicated slide basically just has 2 points, which is that for the vaccine, which is in the blue curve, that the sequence of the viruses from the vaccinated patients essentially have immune signatures that are related to these 2 areas, the 169 and 181 positions, and they are statistically significant, i.e. that vaccinated patients are more likely to have one of these immune signatures than compared to placebo. And it really is at these 2 amino acid sites. Bart then went back to the sera from RV144 and fished out monoclonal antibodies directed to the V2 envelope. Showed the crystal structures of these monoclonal antibodies actually functionally were directed at site 169, and that these monoclonal antibodies mediated ADCC against RV144 trial breakthrough envelope target sequences in a manner that depended on a vaccine match at the site 169. So, you know, you really have this sort of, you know, fishing out on a genetic level and then going back and sort of saying, hey, do we have functional immune responses at this level? And do they have some function in an assay? The answer is yes. Susan Zola-Pausner then showed that the binding of a lot of scaffolded proteins, essentially from many different HIV subtypes, including subtype C, were again significant correlates of risk. So this is not a strain-specific phenomenon. In fact, the strongest scaffold, which may be delusional for us, is in one of the strains we're using to immunize in the subtype C. We hope that that will be useful. And we're moving forward in that way. So, as some famous U.S. Defense Secretary stated many years ago, we do not know what we do not know. Sometimes we do not know what we know. We are not, are these the true mechanistic correlates of protection? The answer is, of course, we don't know this. However, there are circumstantial data are reasonably and strong and form the basis of the planned efficacy studies that the HVTN, as sort of the outlet of a lot of stakeholders in the HVTN in the HIV vaccine field, are designing in what has been called the P5 partnership. And okay, so next part of the talk is sort of saying, okay, what's happened in the last 5 years? Probably for me, the last year and a half or 2 years have been my most frustrating in my 13 years of leading the HIV vaccine trials network. But I'm going to go through those and sort of tell you where we are. So post RV144, which was in September of 2009, we then led a series of thought exercises, which took about 8 months. To sort of say, okay, what do we as a field do next? And there were two major conceptual drivers of this. Number one was the fact that, every time we did an efficacy trial, we got surprised. And everybody's biases were wrong. So we first did GP120, and we thought that it would work, and then we did the STEP study, and we all thought that was going to happen, and not only did it not work, we actually had increased acquisition, and then no one, 20 eminent people, wrote an article that the RV144 trial should never have been done, published themselves in Science. A lot of them are regretting that revisionism, but it's out there. And yet, here's the trial that worked. So it's efficacy trials that really drive this field. The preclinical models and the predictive models, or us as scientists, getting together and sort of say, hey, we think this is it, that has not worked. And that number two is what did work was this POX protein combination, and that we should actually look at that and sort of use that as a basis of continuation. And I would say the HIV vaccine field has been one in which efficacy trials are occurring. Unfortunately, RV124 was started in 2004. It was a huge trial. It took 5 or 6 years to run, from the beginning of the trial to us getting the results, and then getting the correlate stuff was, about 7 years. So if we were going to do one trial every 7 years, this is not the pace that's commensurate with trying to solve the problem. Peter was up here saying, every year of delay, is real. So we need to change how we behave as a field. And number two is we actually need to sort of change the way we do things. So even when we do efficacy trials, we actually really shouldn't be swinging for the fences and deal with home runs. We need to sort of play small ball. Do test of concept trials, to do them as quickly and as efficiently as we can, weed out the crap, try and identify something that's useful, and, you know, and recognize we're not going to hit a 95%. The likelihood of hitting something at 95% is not going to be high. If it is, it's wonderful. We'll be surprised. It's great. But, you know, we need to actually look at this, that we'll go, you know, hopefully we'll get to 50% or 40% or 50%. Then from 50%, we can look at what we should do next to try and get it to 70 or 75%. At some point in time, you can identify what's good and decide what to do next. So essentially do what we call, the cancer field has started calling adaptive design trials, trying to do sequential adaptive trials, in which we are looking in the end of trying to define correlates of risk. And if we do it in the same population and we have two different approaches that give you the same, that have efficacy and they give you the same correlate, well then you probably have an underlying, a really great marker and or maybe the underlying essentially mechanistic correlates. So that's sort of what we talk about, is that you do a series of trials. You look at, rather than one trial or the other, you do continuous monitoring, and if you get a positive signal, you adapt that trial to either enroll the population or not. So, okay, so all right, that's the process. You know, what are we trying to achieve in that process? Number one is if much of the immune correlates at the moment are associated with antibody responses, that doesn't mean it's all antibody responses, but our sort of the biomarkers at the moment appear to be more antibody driven than anything else. So how do I, you know, improve my antibody responses? How do I increase the depth and or the durability? Well, from a durability point of view, adjuvants are one way of dealing with it, and improved immunogens is one way of dealing with it. So we sort of had an approach within 8 or 9 months. And the approach was, okay, we should actually take this POX protein combination and develop and essentially start looking at it in the population, that A, we could do these iterative trials, which A, meant that you need to have reasonably high incidence. You're not going to do a trial of 25,000 people that had an incidence rate of 0.5%. So A, go where the vaccine is needed, so sub-Saharan Africa. And B, there was every indication that you were going to need clade-specific immunogens, and redo the immunogens so that they were clade-specific. And a group got together who, A, were involved in this, NIAID, the Bill and Melinda Gates Foundation, the HVTN, Sanofi that made the LVAC vector, and brought in Novartis, mainly from their acquisition of Chiron, looking for a pharmaceutical partner that would agree to, if there was success, actually make GP120 in the kind of quality and production that if we were successful, could be brought into the marketplace. So by the end of 2010 and 2011, we actually had, essentially by January of 2011, we really had a clinical trial development plan. In which we would manufacture an LVAC vector that would be similar construction and backbone as the LVAC used in RV144. We can use a clade C strain. We were going to make a bivalent GP120, and these strains had been picked. One is a sort of an older isolate, TV1, that was isolated from an acute disease patient many years ago, and one 1086 that was actually one reasonable from a primary infection founder strain from a Capriza isolate in KwaZulu-Natal that was isolated, in the probably 2009 region. They were selected for their manufacturing ability, and they passed all the issues of binding to V2-based monoclonals. They listed V2 loop antibodies, and the products were then decided upon by a series of committees and commissioned to start being made. And I will say the frustrating part has been that that it has taken a long time to manufacture the products. And we've had a 1-year delay by Novartis in actually manufacturing the GP120. And really it will be over 5.5 years from the time of knowing that we had GP120 to actually starting the efficacy trial. Now, we will be starting the phase 1 trials in the end of 2014, but that's actually 5 years. And I have to say we need to somehow look back at this and sort of say, why is it that we are taking so long to make products for vaccines? When you identify, something that could work from an HIV vaccine field. It is, we still have not repaired some of the structural deficiencies in how you do research in HIV vaccines. And I'll stop, maybe someone will ask a question about that. I took my Pepsid this morning, so, I can answer it maybe. We will start phase 1 studies. And the first thing we're actually going to do, is take the Thai vaccine that's been sitting in the freezer. So the stuff that was used in Thailand, not clade matched, it's the AE, and actually give it to South Africans. And actually sort of find out, are there immune responses similar to Thai's? One of the reasons we're doing that is actually one of the real interesting things that we've been picking up lately is there's been a good correlation between BMI and immune responses, especially T-cell responses. And South Africans are heavier than Thais. Women are lighter than men. If you look at a lot of vaccines, especially even in the Thai trial, women respond better than men. And it's an interesting sort of observation that has come up maybe with sort of partial, not vaccines that are as immunogenic as many of the vaccines that we have. So we want to actually get the immune response sort of that allows us to get some assurity that we will be able, that the genetics of the population is actually not going to alter the immune response. So we really will have a very good fix between South Africans and Thais as it relates to that vaccine. Then we actually start the real work as soon as the stuff becomes available, which is a phase 1 study of the LVAC vector with this bivalent GP120. And so this is called HVTN100, which is a sort of a qualifying trial. In other words, we have some targets of, we expect the vaccine to be immunogenic, but I will also tell you that there is not even the non-human primate, is there really predictive efficacy from what you find, immunogenicity, not efficacy, from what you give a non-human primate versus what you give a human. There are many examples in which we have a lot of immunogenicity in the non-human primate and not much in humans. One really great one recently, and I'm not going to show this, has been in a DNA vaccine that the Penn group has done by electroporation, in which they gave the exact same vaccine. This is an HIV-1 vaccine that was given to non-human primates, electroporated in, has an envelope construct, and the immune responses to the envelope were 50% of the response in the non-human primate. We gave that vaccine in humans, and they had no response at all. They had great responses to GAG and PAL. By electroporation, we actually got DNA responses and CD8 responses, CD4 responses. Electroporation is really, neat for it's really an advance for DNA vaccines, but absolutely no OMV responses. Whereas it was 50% in the non-human primate. So our ability to predict and use these preclinical models is really not very great. And Greg, I'll show you the slide if you want. We need to qualify the vaccine and show that we can elicit the kind of V1, V2 antibodies and the kind of binding antibodies that will allow us to do a correlates of protection study. Because if we can't do a correlates of protection study, we shouldn't do the study. That's what this is all about. We need to qualify it. And then we move to an efficacy study, which is planned as a 5,400 patient efficacy study. Now, I do have to say that despite what I said about sequential adaptive designs, both the South African government and the manufacturers here wanted to do a potential licensure study. It's the issue from their point of view, and it's not an unreasonable point of view. They weren't in this business to just do science. They were in the business to try and bring a product to licensure and to distribution. And actually, the South African government has agreed with that, and they actually are a co-funder of this trial, and that's one of the reasons we're going ahead and doing it, is to listen to your partners and not just do what the NIH wants to do, or even the Bill and Melinda Gates Foundation wants to do. So we are doing, this is actually more of a classical, I want to hit a home run, and the South Africans feel that's what they want to do. But we also split off the sequential adaptive design trial, and we are looking and taking ALVAC and putting it into the phase 3 program, and taking the other poxvirus, what we'll call the second generation poxvirus vectors, and putting that into a sequential adaptive, what we call a research trial. We're using NIVAC, which is a product also developed by Sanofi. It does have greater transgene expression than ALVAC. It does give greater recall memory, CD8 and T cell responses, and induces cross-presentation. And the non-human primates essentially has greater immunogenicity than ALVAC. It is quite a different, NIVAC is based on vaccinia, and ALVAC, of course, is a canary pox vector. NIVAC is a vaccinia-based vector. It is different in its genetic genes from MVA, you could say, well, why aren't you using MVA? Well, it is because the Bill and Melinda Gates Foundation through Jeppy Pantaleo has been working on NIVAC for the last 7 or 8 years as a backup vector to ALVAC. There have been some comparative studies with the clade C products, comparing an ALVAC clade C with the NIVAC clade C in non-human primates. I'm not sure this is projecting that well, but the A's are the ALVAC and the dark are the NIVAC. They both give pretty equal binding antibody responses, but T cell responses are are much better with NIVAC than ALVAC, about double. And if you also do a DNA prime as related to NIVAC, you markedly increase the T cell responses. So this is a non-human primate study, which in the left, you just need to conceptually see, hey, this is hugely different than this. This is NIVAC alone. This is DNA NIVAC. So just logarithmically better CD4 T cell responses with DNA NIVAC. Now, one of the interesting issues is and this has been true with all poxvirus vectors. So DNA prime, NIVAC, a DNA prime, a heterologous prime and boost will give you better T cell responses. But for the poxvirus vectors, I can continue to give that poxvirus vector many, many times. You develop antibodies to the immune responses to the pox vector with multiple immunizations, which cut down your T cell responses, cut down your neutralizing antibody responses, but your binding antibody responses get boosted by continuation of those NIVAC vectors. And actually, this is an interesting slide that actually compares the V1, V2 antibody titers, and I could show you the ELISA antibody titers, between multiple doses of NIVAC versus DNA NIVAC. And so, in the end, they'll get to the same, but from a time course point of view, you induce much higher levels of binding antibodies by actually giving MVA more frequently. So it's going to be an interesting issue. So DNA MVA versus multiple MVAs, or DNA NIVAC versus multiple NIVACs will give you different immune profiles. And it'll be interesting as to what goes forward. So what we are looking at for the research study, the adaptive design study, is a trial that looks at NIVAC alone and DNA NIVAC alone as the vectors, uses GP120 as the boost, and we're thinking about trying to induce people to do a factorial design in which we use actually different adjuvants. And the adjuvants we're trying to look at is, number one, Novartis's MF59, which is, licensed in many places of the world as a, sort of Th2 tropic antibody-producing adjuvant, versus GlaxoSmithKline AS01, which is much more of a sort of a Th1, I hate to use these terms, adjuvant that shifts the immune response in a slightly different way. And the strategy here is really to try and get diversity in immune response. All right, so the more diversity we have, the more we will learn from a correlate's point of view. And that's the strategy that one is looking at. Praying for some efficacy. Ideally, the best efficacy you would like. If you can't have 90% efficacy, you really want 50% efficacy, because that really allows you a huge power to do the correlates analysis. What else is going on? We're going to stop there, 5 minutes. So that's really the essence of the strategy that's going on, sort of, I guess, worldwide as it relates to the post-RV144 story. There are, as you heard from the earlier groups, many other vaccine immunogens in the pipeline. I'm happy to ask at the end of the next series of slides where, what's going on with DNA vaccines, this vaccine, that vaccine. But, it's probably not as important as the, other than the story of what's going on with RV144. So I'm happy to answer some questions, and my pleasure. Thank you.
Question there? So, Larry, great talk. Thank you very much. Yesterday at our workshop, we discussed very, very briefly the idea of placebo-controlled trials. We discussed very briefly the idea of placebo-controlled trials and whether ethics committees moving forward are going to permit full placebos in the light of results from PrEP and so forth. Can you give us your views and comments?
Sure. Well, we've spent a lot of time on the issue of PrEP. First of all, I don't think there's any problem with placebos as it relates to vaccine trials, and even in our phase 1 trials, in Africa as well as in the United States, we do have placebo groups. We weight them. All our phase 1 and phase 2 trials are sort of randomized blinded trials. Usually it's 80% vaccine and 20% placebo, and that is because, especially as we do exploratory assays, we need to know what the background is, and we also think it's good to, it allows us also to, after phase 1, to compare one vaccine with the other and make some judgments on clinical development. So we've not had any issue in any placebo control, the use of placebos for vaccine versus placebos in whatever country we do. So then the next question you're asking is, okay, what's the role of PrEP, how we view it through the vaccine fields ours? And we've had a lot of discussion about this. Of course, we have an ongoing phase 2B trial of a DNA Ad5 vaccine from the VRC that's going on in the United States at the moment in the MSM population. As soon as the IPREX study came out, we did an enormous number of focus groups and stakeholder issues about how to handle PrEP. We even arranged with Gilead that for people who want PrEP, we will, through a central pharmacy, provide it for them free. And I would say to you, at this point in time, we're monitoring it. We haven't done drug levels, but by self-reporting, after a year and a half of IPREX, less than 5% of the people who are on HVTN 505, which is now fully enrolled at 2,500 people, are self-reporting PrEP. And this is an MSM population in the United States, and the sites are, include New York, in San Francisco, Seattle, et cetera. So, the disappointment of the recent studies in South Africa and sub-Saharan Africa with microbicides, You know, we had studied, again, we were having a lot of focus groups and a lot of discussion with the South African investigators and authorities in making the decision as to how they feel about PrEP at this point in time. I think there's the sense that we will not need to use PrEP in these vaccine studies. We certainly will do counseling. We generally approach this in a way of having the investigators in the country that we are working in decide. And we hold an extensive series of stakeholder engagements that I think the HVTN is very proud of its global community advisory boards and the people who are involved, and we pay a lot of attention to that. So, I mean, that's our, you know, it is a dynamic field. If the data changes, we will react to the data. We're not afraid of doing combination studies in any way, shape, or form. There are many reasons to believe that that an antiviral would decrease the burst size and increase the efficacy of the immune response in controlling that. And so in many respects, you know, I put on my science hat and they should be maybe even more than additive. They should be synergistic, and okay, they make it harder to do the study because you'll decrease the incidence rate and therefore you need more people. But, you know, you should never be, you know, but we're certainly not afraid of the science. And so if that's the way the people you know, feel they, and the field should be, we'll follow it.
One more question here.
I was curious about your description of the 097 study in light of the information you described about the Sieve analysis. If there's really something about the virus that you're exposed to in signaling a difference in the efficacy level, would that affect the design or the utility of using the same product in South Africa?
Well, we're using the same product in South Africa only in a small phase, you know, I guess you'll have to call it a phase 2 study, but we're going to do it in low-risk people. This is not an efficacy study, okay? So we are just wanting to know when we, as we move towards true efficacy, if we get differences between what we see in South Africa in efficacy versus Thai in efficacy, that we will actually be able to know what whether this is due to the immune response differences to the vaccine itself or to the genetics of the population itself. And so taking the same vaccine and knowing what it did in Thais and comparing it to what it does in South Africans helps us with that. Well, whether we'll be clever enough to figure it out if there are differences is another story, but we will have the samples and have done the experiment.
So we have one more question, then we'll have to close.
Okay.
So Larry, I was fascinated by the IgA story, and as you know, there's been a lot of interest in trying to understand better how much we should be augmenting mucosal immune responses. And you didn't comment on whether they looked at secretory or non-secretory IgA. My understanding is that was non-secretory IgA that was, or systemic IgA as
Correct.
Opposed to, so that in fact this would not be anything to do with mucosal immune responses, is that correct? Okay,
Correct.
so that's an important
Unless there's
distinction.
some surrogate, unless they're linked in some way. But you are exactly right. These are serum IgA responses. There were no mucosal samples done in the Thai study, and we're going to think about getting some in some of these studies. Thanks.
Thank you, Larry. If
Thanks.
you want to stay here for a moment, I'd like to have the 4 PI presenters also come up to the stage so we can give them all a big thank you. And a little token of our appreciation. Larry, for your great oversight into complexity to no end. Right?
Well,
thank you very much.
Oh, thank you. It's a pleasure.
And also to all of our other PIs. Peter, thank you. Jerry, thank you very much. Good work. Thank you very much. So that brings us to the end of this session, Vaccine Research Plenary. Thank you very much. Enjoy the rest of the day. I think we've set the scene very well.